much. If there is anyone who wants to add more into it regarding chemicals that we are using in this whole experiment, anybody wants to add something? Pratna, would you like to add something on it?
We use only two chemicals.
Okay, what are those?
Alcohol and distilled water.
Okay, so can you tell us why this experiment is important? What is the use of this experiment? What learning is actually you have got to know after doing this experiment? What has been the main learning process?
Know about the different pigmentation
Okay.
contained in a screen, like yellow, green, dark green.
Right. Colors and all? Right. So thank you so much for speaking with us, and we wish you all the best. Keep doing what you're doing. Thank you so much. So now we are in the second deck, and the boys are students of class 12 science actually are conducting this chemical experiment. So we would have a word with them and try to know what experiment they are conducting, and as compared to the experiment that we just covered now, it looks quite different. So let's know what is the basic difference between. So hello guys, what are you doing here?
Fine, sir. We are doing titration.
Acha.
From this, we can find the molarity and strength of the given solution.
Okay.
Here we have KMnO4 solution,
Okay.
and which is of known molarity.
Right.
We know its molarity, and here we have oxalic acid solution.
Okay.
This is unknown. So
Alright.
we have to find the molarity of the solution. And so many solutions and chemicals are there in our nature that we don't know the molarity.
Okay.
But by this method, titration, we can find the molarity and concentration of the given solution or any of the chemicals present in the nature.
All right. So if I may ask, what chemicals are we using to conduct the experiment?
Here in pipette we have KMnO4 solution.
Right.
And here we have more salt
All right.
with dilute H2SO4.
Right.
By this we can find the colorization
Right.
of this titration by this titration method.
Okay.
If it turns into pink color, then we can find out from the reading, from the initial and final reading,
Right.
that how much molarity and strength is
So
in
we
the
have to
solution.
observe the initial reading and the final reading, and then we have to calculate and find out the final results.
Okay. So it seems like you all are busy and everyone is putting their best knowledge forward to help one another, and it's a group work and everybody is doing the best they could, and Right.
Strength of the molarity
Okay.
of the solution.
Right. Thank you so much for speaking with us. We'll let you do your job. Thank you so much. So this is the final interview. We are in a last desk and we have a bunch of students who are conducting the experiment. So we are going to speak with them, try to understand what experiment is this, what is the importance of this experiment, and how this experiment is helping them to understand the theoretical knowledge, and how the practical and theoretical is going parallelly to make subject matter more comprehending and more comprehensible to everyone. Okay, so guys, hello everyone. How are you doing today? The first thing is.
Yes, so we are all doing fine today, and it's the present weather, and we are all like feeling very excited for chemistry practical
Right.
examination, which we are like practicing right now. So like talking about my experiment, so we are performing titration right now.
Right.
So like giving a brief gist about the practicals. So like here we have the curate, this long thing, and in this we have used two chemicals, that is oxalic acid, and the second one is H2SO4 solution, that is
Okay.
diluted H2SO4 solution.
Okay.
And in this we have the mixture of both the things. And in this the purple color solution is KMnO4 solution.
All right.
And so like we can see that there has been a color change in the solution.
Okay.
Like we can see a faint pink color in the solution, which means that it has been successfully titrated. So like talking about titration, like it helps in determining the strength and the molarity of the solution. So I would like to ask my friend to explain about the molarity.
Right, so we would definitely like to understand what is molarity and the titration as a whole. What is it? If you could walk us through and shed a light upon it.
So molarity is defined as the number of moles by volume of solution later.
Right.
So by this titration we get to know that what is the strength of this molarity of this KMnO4 solution.
Okay.
So first of all the like auxiliary acid solutions and digestible are taken. These are unknown. So we calculate this solution by titration. We are calculating this experiment, but we are getting the molarity of the KMnO4 solution. Like we are